So it's a real pleasure. Ahmed was one of our earliest students in our PhD program. So he is a graduate of the PhD program in biomedical and translational sciences, and I chaired his thesis committee. And when he first introduced his research at his first dissertation committee meeting, he started sharing his ideas on what he wanted to propose for his research, and I was blown away. I remember asking Ahmed, I said, Ahmed, is this all your own idea? And he said, yeah, this is something that I observed, and I thought this would be worth looking at in the lab. So really exciting stuff on implications in delivery, in diagnosis, in tissue repair, and we're trying to lure him over to the dark side of cancer. I thought it was also really interesting, with the Heart Institute, the work being done with cardiosphere-derived cells is really something unique. I mean, this predated iPSC-derived models, and these are still useful and important systems to work with. My lab has been working with your cardiosphere-derived cells, and they've come in really handy. Now, since graduating with colors and honors, Ahmed now holds a faculty position, and I have been on his students' dissertation committee. So I don't know if I should be feeling like I'm getting old or you should be feeling like you're getting old. In either case, though, it's a real pleasure to see the circle continue itself. And I am really pleased to invite Ahmed over to our community of bioengineering ideas. So I think the topic is really timely. One last thing that I'd like to say is that this is the first of at least 3 exosome-related talks that we'll be having in Tribe. One related to cardiosphere-derived cells. We will have an upcoming talk having to do with exosomes derived from cancer cells, and then another one having to do with exosomes from placental tissue. So I think that we've got a really neat theme going on, and that takes advantage of some really unique aspects of Cedars-Sinai. Awesome. So without further ado, take the floor.
Thank you. All yours. Wow, that's way better than I had in my biography. I will say one of the biggest sort of appeals for Tribe is knowing that Lolly is involved, and Lolly has been not just a prolific scholar, but a leader and a mentor of mine. So I always appreciate you. And anytime I have a student in the lab, I say, you have to have Lolly on your thesis committee, because she will give you ideas and she'll make you better, and she won't hurt your feelings in the middle. And I've been interested in cancer for quite some time, to be honest. My first job in college was to work at a cancer database. And this was before, machine learning and all these things. And I would read PubMed papers, and I would synthesize and summarize under every sort of clinical or cancer target. And that was my job. And so I've always been interested in cancer. And I'll be honest, when I started the PhD program here, even though I was very much interested in cancer, it scared the hell out of me. And the reason why is because I've seen one target come and go after another. And I always wondered, are we ever going to get to a real cure? Because everything looks rosy and beautiful in an article or a publication. But then 5 years pass by and you hear about, nothing ever happens with glioblastoma or pancreatic ductal adenocarcinoma, and so on. And so out of sheer cowardice, I decided, well, let me go somewhere easier. Maybe regenerative medicine might be better. But my heart has always been interested in cancer, and I feel like a lot of physiology origin fields owe a huge debt of gratitude to cancer, because we're not very good molecular biologists, quite frankly, and the pathways that have been identified in biology have been incredibly helpful in understanding phases of injury and acute inflammation and fibrosis and all these things and remodeling that have really helped push our field forward. So there's a huge debt to be recognized from the cancer folks. And with that, we'll start. So It's going to sound a little hippie in the very beginning, but it's going to get more specific and relevant. And so when I say in the title that the cells speak for themselves, I'm talking about a very conserved language across biology, and that's extracellular vesicles. And I'm happy to hear that there's more than one EV talk coming up, because it's just something that's really unmined and has a lot to offer, both from therapeutic perspectives and for emergent discovery. So today I'll be talking about the rationale of exomer drug development. Why is it important? Why is it unique? What does it have to offer? And then I'll talk about the CREDO pipeline. The term CREDO was coined by my dear mentor, Dr. Eduardo Marban, and it describes really what the discovery pipeline looks like. And I went back and forth about what to talk about. I could have delved into one exomer drug, one RNA drug, but I decided to give you 3 vignettes, if for nothing else, to keep everyone's attention captivated. And then I'll talk about the other exomer drugs in the pipeline, because it is, after all, a pipeline. There are many candidates. And finally, I'll talk about its relevance to cancer drug development, because that's something that is near and dear to my heart, and that's something that we're beginning to get our feet wet with. But first, I want to talk about what EVs are. So these are lipid bilayer membrane particles that are secreted by all cells. And when I say all cells, I mean every single cell, every self-respecting cell that calls itself a cell. From the most modest archaea to the most complex neuron in the cerebral cortex, every cell secretes EVs. That's important because when you think about the interface between the biotic environment and the non-biotic environment, you realize that EVs are everywhere. They're in blood, every body fluid you can think of. They're in every juice that you can think of. They're in honey. They're in milk. They're in the ocean. They come from bacteria. They are everywhere. And to understand how the world works, you can't ignore that very important interface where biology speaks to the abiotic environment. And from a mechanistic standpoint, there are 2 main types of EVs. Of course, the good folks at ISAB will tell you that there's 50, but there's really 2. There's one where there's a very deliberate pathway of packaging through the late endosome. And then with progressive acidification, you get release of these rather smaller particles. And then you have the passive membrane shedding that are called ectosomes. And there are multiple classes under each, but generally speaking, that's where they come from. Now, just to give you some perspective, I know when we think about biology, we think about medicine, and we think about the human body. But just I want to talk about the broad sort of scope of EV science. And as you can see, EVs are involved and they play critical roles in every aspect of human health and disease, from conception all the way down to aging and senescence. But more broadly, they are mediators of communication across different relationships of organisms, from maternal to fetal, to the bacteria as they communicate with the epithelium and the immune system, from plant pollinators to bacteria and fungus communicating EVs through root systems, through parasite host, virus tissue, and predator prey. They truly are nature's most effective mode of communication, and that's why they've been conserved for all these years, and they have multiple functions. And in understanding those functions, we can begin to try and hypothesize how EVs work in the context of different diseases. It's not simply exchange of communication, but it's also used as a defense mechanism. So, for instance, cancers will secrete EVs that have decoy receptors that will sequester chemotherapeutics. For instance, there are also food sources. For instance, bacteria will secrete EVs that sequester nutrients in the gut, digest them in the lumen, and then are uptaken later. So these multiple functions need to be completely understood before we can begin to think about how EVs fit in the disease model of choice. Now, for a long time, there's been this misconception that all EVs are the same from any different cell type. But more and more, we're realizing that EVs are as unique as the cells themselves. What you see on the right, on the left, are the RNA distributions of 2 different cell types. And they couldn't be any more different. One is the CDC EVs, and cardiosphere-derived cells, as you know, are these stromal-derived cell therapies that have passed phase 3 clinical trials and now under BLA consideration for Duchenne muscular dystrophy. So they are patently therapeutic tissue reparative cells. And then you have EVs from regular old skin fibroblasts that are therapeutically inert and do nothing. And you can immediately appreciate that just from an RNA distribution perspective, they're very different. And the conceptual framework that I like to think about when considering EVs is in the form of language. They essentially do the same thing, which is communicate information. But the type of information and the language used and the dialect used is so context specific, all the way down from two cells of the same type, but under different conditions. And that's important to think about when we think about, again, EV research. And then comes the major opportunity that exists in this field. It's interesting to note that 98% of our genome is non-coding, much of it being regulatory. And that means that this 98% is regimented towards regulating that 2%. That's huge, especially when you think about the fact that the druggable proteome is only 15%. I had a meeting with Josh Sassine, you all know him, he's a good guy, and we were talking about what the big challenges are with cancer. And he said it's not about understanding what the targets are. We more or less know what genes the tumors are addicted to. We just can't get to them. And in looking at the different RNAs that exist, maybe there's an RNA out there that can reach these targets where a protein or a small molecule inhibitor can't. And to me, that's the big allure of what we're doing. And so therefore, the rationale for exomer drug development is decoding EV-derived small RNAs, much of which we know nothing about. And that's the best compartment to look at, because if cells are trading this between each other, that means that they must mean something. And that is perhaps the richest catalog of bioactive RNAs, where we can begin to discover how they work, gain mechanistic insight, look at disease differently, and then maybe at the end have a therapeutic candidate in an indication where there has been historically an impasse. And this is graphically depicted here. So we take a population of therapeutic EVs, meaning they come from cells that are tissue reparative. We understand how they work. Actually, before we do that, we confirm that they're biologically active, because who cares if they're not, right? And then once we understand what the mechanism is, how does this RNA impact the injured microenvironment, then we use it as a template to develop an RNA drug. We test this drug for bioequivalence, and then the last step, which is non-trivial, is to find a disease indication. Eduardo and I have had countless discussions about what's the best indication for a certain RNA. Now, certainly there are considerations related to regulatory issues. It has to be, a miserable disease with no cure, rare disease even better. There has to be a solid animal model for it, and so on and so forth. But we have to match the mechanism with the indication. And I'll show you, albeit briefly, how each of these RNAs have been matched with a fitting indication. Now, what's an exomer? Well, as I said before, an exomer is a synthetically designed or synthetically made RNA that's based on an RNA that we discovered in EVs. And they have some similarities, and they are different in others. The biggest similarity, of course, that they're EV derived, but also, and perhaps inexplicably, they all seem to signal to macrophages. And frankly, we don't know why. Eduardo and I have had discussions about whether it's because macrophages have first dibs on anything that's injected, or perhaps the EVs that are coming from the cells that we've been studying naturally signal to macrophages. We don't know. But the corollary to that is that they're all orally bioactive. And the reason why I say that's a corollary is because when we've done biodistribution studies, when you feed these animals the RNA and the formulation that we have, they all seem to be preferentially taken up by the gut-associated lymphoid tissue, including the macrophages of the lamina propria and the Peyer's patches. The only other time where you see them is in PBMCs. They're not even swimming freely in circulation. And that tells you that there's a very deliberate exchange there somewhere. And I think to myself, if these macrophages were not RNA active, I wonder if they'd be active at all when orally delivered. But it just so happens that this is the case, and it's quite fortuitous. Of course, they're all different because their genomic origin is different, their oligomer size is different, their gene targets are different, the mechanisms by which they affect these gene targets are different, even the effective dose which kind of makes sense because with different kinetics comes different effective concentrations. But essentially, exomers are all of these things. So let's talk about the first vignette, and that is CDC EVs and the TY1 exomer. Now, I've already told you about what CDCs are, so I'll just move on. We discovered two fragments that are very similar, one larger than the other. The first one is called EVYF1, and there's a smaller variant that we call NT4, and it derives from the human YRNA4 gene. Now, we don't know much about YRNA genes in general. We know that they stabilize DNA during replication, and some of them are involved in switches between metastasis and tumor silence, and some of them are also involved in autoimmune disorders, but by and large, we're not sure what they do. So we discovered these two fragments, and we thought to ourselves, if we're going to look at something effective, it's better to look beyond microRNAs, which the literature will tell you are very sparse and few in EVs, but these are actually quite abundant. The first thing that we do is we look at our in vivo bioactivity model, and that's a 48-hour ischemic perfusion model. So we take a rat, we tie off the coronary artery, we wait for 45 minutes, and then after 45 minutes, we open it up. Perfusion happens, injury happens, and then 20 minutes into perfusion, we give the animals the treatment. So we gave them NT4. Now, of course, all the RNAs that I talk about when given IV are formulated in a lipid nanoparticle. So just to get that out of the way. And what you can immediately see is that NT4 shrinks the size of the infarct 48 hours after injury. So this is the green light for us that this is an RNA we're studying. I won't belabor the structural activity studies that we've done to arrive at this structure. But we did two things to this NT4 sequence. One, we introduced a mutation, a GDC mutation, the 5' end. And to enhance stability, we introduced, through trial and error, 3 locked nucleic acids alternating on the 5' and the 3' end. So there's 6 locked nucleic acids, and thankfully that did not affect structure. And once again, we confirmed that it's bioactive. Now, how does it work? Now, there's a whole paper dedicated to this mechanism, but we only have an hour, so I'll be very brief, and please let me know if you have any questions. We ran a sequencing of macrophages, because again, these are all macrophage active RNAs, and we found that the one pathway that popped up the most was this DNA damage response. Now, before then, we'd never thought about DNA damage response. All we thought about really was inflammation and fibrosis, and not much else. But this caught our attention, specifically the TREX1 CGAS sting pathway, which is a very primordial pathway for detection of damaged DNA, probably from back in the days when bacteria had to wrangle with bacteriophages trying to inject their DNA into them. And then we wanted to figure out what the binding partners were, and we took a two-method approach to find out what it binds. And the gene that came up in both was a gene called TREX1, which you've seen earlier. That's a 3' exonuclease. It's an enzyme that degrades leaked DNA, double-stranded DNA in the cytoplasm. In the nucleus, it participates in the complex that is involved in DNA repair. So we found that with TY1, you had an increase upregulation of the expression of this TREX1. And then when we did binding studies using a luciferase assay, we found that it binds the 5' UTR of TREX1, which is reassuring, because often small RNAs that activate or enhance the transcription or translation of mRNA targets tend to bind the 5'. siRNAs, microRNAs all tend to bind the 3'. And when we looked at the DNA damage disposition of these these heart tissue, 48 hours after injury, you find that indeed TREX1 is up, and that in data that you see to the left, that the phospho-HOAX is a marker of DNA damage is reduced. And so that is the summary of how TY1 functions. They function again by signaling to macrophages. A stress trigger happens, inflammation, tissue damage, whatever else. TREX1 is uptaken, DNA damage goes down, immunomodulation occurs, and then from that happens tissue repair.
Can I ask you a couple of
Yes.
simple questions?
Please, yeah.
Where it comes from?
The NT4? So we discovered it in the EVs of these cardiosphere-derived cells.
So carbon.
Yes.
And when you say abundant, what do you mean? And when you do the experiments to demonstrate effect on DNA damage response, what's the relative concentration? Is it equivalent to what you see in serum, et cetera?
That's an excellent question. We've never actually looked at NT4 in the serum because of the animals that we work with. They don't have this NT4. We've never tested NT4 in human serum, which is probably we should. But you raise a really important point, and that is the number of copies of NT4 in CDC EVs are on the order of 3 in every 100 particles. Allie's laughing because I looked at her and she's like, I don't know. But it's 3 copies per 100 particles, which is far less than the dose that we inject. What we inject is 0.2, well, for IV, it's 0.15 microgram per gram, or mg per kg. The question is it because better delivery with the EVs or what's happening? At the end of the day, if they go to macrophages, you have the amplification mechanism, which may obviate the initial dose, but it's a good point. Right, now comes the job of figuring out what is a fitting indication. And what we found is that there's this disease called systemic sclerosis. Actually, Eduardo has been thinking about that disease for a long time, since his days in residency, because he came across a very memorable patient. But it is a disease where the immune system attacks connective tissue. And what happens is you have progressive thickening of the skin, you lose articulation in your fingers, the mucous membranes begin to swell and thicken, and then you have internal fibrosis. Most patients will die of respiratory failure, and this right behind that is cardiomyopathy. So it's a disease that is very highly inflammatory with a long fibrotic tail. 2 things that we think TY1 can enhance, can improve, because its underlying effect on DNA damage. Okay, so what I'm showing here is a model of systemic sclerosis. These are mice, wild-type mice, that receive subcutaneous injections of bleomycin 3 times a week. After 4 weeks, the animals have full-blown pro-inflammatory and fibrotic features. And then when we feed them TY1, as you can see, the skin thickness goes down, which is a huge clinical indicator. And shown here, this is the blue segment, that's the skin thickening that you see, and that you see it attenuated here in TY1 fed animals. They're fed TY1. And you also see the same manifestations internally with reductions in cardiac fibrosis. Now, I'm not showing you all the readouts, but there are improvements in diastolic dysfunction, in exercise capacity, and others. I can point you to the publication that demonstrates all this. So the next vignette comes from another RNA that we discovered in immortalized CDC EV. So quite a while ago, we tried to figure out how we can make CDCs immortalized. And that was not an easy task because it meant that we needed to understand what made them potent to begin with. 5 years later, we understood that it's canonical when signaling. And the way that we immortalize them is that we use traditional methods like the simian virus, small and large C antigen, along with a lentivirus against this gene that regulates canonical when signaling called MEST, or PEG1. It's an imprinted gene. In any case, we isolated the EVs from these immortalized CDCs, and we call those IMEX. We sequenced them, and then we found that there were 2, according to sequencing, they were indexed as pi RNAs, and that gave us a hint into their function. But the broader scientific community could not accept the fact that pi RNAs were at all functional outside of the germline. And so we were beaten back, and we had to reconsider what these genes might be. So we did a span of where they showed up in the human genome, and we found that they also had hits in the YRNA4 gene, which is interesting. It's actually not the 5' where the NT4 comes from, but the 3'. And we call that YRX3. Again, we run the bioactivity assay to show that they are bioactive, and it is. And because of the fact that it was a pi RNA, we were able to sort of glean some hints, and we did bisulfite sequencing to find out, to try and find out where the function was, or what genes it was methylating. And this gene called PIC1, which we'd never heard of before, Turns out that WireX3 binds the 5' UTR and intron 1 of that gene and causes methylation. And without the gene silenced, and then with further mechanistic studies, we found that SMAD3 phosphorylation also went up. Now, SMAD3 phosphorylation is involved in enabling macrophages to be more erythrocytotic. They become Pac-Men. They're better able to gobble up debris. And yes. And this is the figure on the right shows that macrophages that were either incubated with WireX3 or down of an siRNA against PIC1 are better able to eat up dead cardiomyocytes, which verifies our hypothesis. And then on the left, you see widespread SMAD3 phosphorylation in the sections of animal hearts that received WireX3. And so with that, we found out that TY2 is a drug, but NT4 enhances PIC1 suppression or triggers PIC1 suppression, and then enhances SMAD3 phosphorylation, allowing the macrophages to be prophagocytic and better able to clear injured tissue. Then we decided to develop a drug, and similar to what we've done before with locked nucleic acids, we added locked nucleic acids to this peptide, or this oligomer, I should say, and then we introduced a U to A mutation on the 3' end. And again, we looked at the bioactivity, and it was bioactive. And we looked at several disease models, including sepsis, because it makes the most sense. Sepsis is where you have bacteria just, overgrowing, and they're in the bloodstream and they're in tissue. And we just recently published a paper in JCI Insight about the effective role of WireX3 in enhancing macrophage clearance of bacteria in tissue and in circulation. However, we're also keenly aware of a disease called arrhythmogenic cardiomyopathy. What you see here is an overblown image of the intercalated, well, it's a model of the intercalated disc, and that's what adheres cardiomyocytes to one another. There are a family of mutations in each of these proteins that compromises this adhesion, and then with every contraction, you have cell damage, you have the release of debris, and that triggers a highly inflammatory phenotype that is really mediated by all that junk that bleeds out of injured cells. And we figured, well, macrophages that can better clear cell debris would probably be efficacious in this model. And indeed, animals that were given, in this case, IV injections of TY2, which is the drug form, the exomer form of WireX3, enhances cardiac function, and this is the left ventricle, this is the right ventricle, enhances the QT duration, or actually suppresses the QT duration, which is good, and then it improves cardiac velocity. So the signal is better able to be conducted across the heart from the left ventricle up, and it also decreases fibrosis. And then the last vignette that I'll cover is one that we are now working on, which is TDO2 EVs. And let me tell you what TDO2 EVs are for a second. So I told you we have CDCs. I told you we immortalized them. The next thing we wanted to do was to create CDC-like cells that we can engineer from fibroblasts because they're more accessible. And this is the work of my dear friend and colleague that we lost recently, Kyle Peck, wonderful, wonderful human being, a great graduate student and a valedictorian in 2022. He had discovered that canonical wind signaling activates the expression of this gene called TDO2, which is a tryptophan metabolic enzyme. And that if you knock in TDO2 in fibroblasts, you get cells that are equally as therapeutic as CDCs. So now we had another EV population that we can scan and look for therapeutic RNA drugs. And this is just to show the abundance that goes back to your question earlier. The abundance of this gene we call URX1. We call it URX1 because it's from an uncharacterized portion of the genome. It just has one of those alphanumerics that don't mean anything. There's no name to it, and yet it's there. It's there abundantly. So we called it URX1. So then again, we ran it through the typical bioactivity model that we use to examine RNA candidates, and indeed, this was potent. And then once again, we made a drug version of it by having a G to C mutation and then including the locked nucleic acids. And what you see here is the bioequivalence of that RNA drug. Now, what the mechanism is and what the indication would be, it remains an active area of investigation. But we found out something really interesting. And we found out that, and this is Hiro's brilliant work, is that it seems to suppress phospholamban in myofibroblasts, which any physiologist will balk at and say, what do you mean? Phospholamban should not be in a fibroblast. It's part of the contractile system of a cardiomyocyte. And yet that's what we see. It's a bit controversial, but it seems to suppress phospholamban in myofibroblasts and really reduce profibrotic signaling. It's exciting because the cool thing about new RNAs is that at the nexus of their discovery is emergent understanding and therapeutic potential. So perhaps it's true, perhaps it's not, but time will tell. And I want to end with, how are we doing on time? Okay, it's 46 minutes. I should leave us some time for questions about the pipeline that we have so far. So there are many other RNAs that are being investigated by my colleagues at the Heart Institute. Some that are fragments of mRNAs, some are fragments of tRNAs, and some are fragments of long non-coding RNAs. The EV secretome is replete with many different RNAs that are just there for the picking and the understanding, and we're so excited about learning more. This is, at the end, a cancer seminar, and what I presented to you in the beginning was one in the context of regenerative medicine, but really what I'm thinking about is tumor EVs are chock full of small RNAs that we know nothing about, and there are messages that are being sent to other tissue. Messages that don't mean well. They mean to invade, they mean to suppress the immune system, they mean to affect fibroblasts to make the tissue more amenable to invasion. If we can find some of these RNAs and develop antisense drugs against them, that would be fantastic, because in the sort of the war of cancers, it's going to be more than one approach to turn that acutely deadly disease into a long-term, chronic, manageable disease, the way HIV has been in my lifetime. And with that, I'd like to thank everyone who's involved. I'd like to thank Eduardo, if he wakes up. he can go to sleep because he's seen this a million times, but who's just been a fantastic mentor and a friend and just a joy the day I worked in his lab. And all my wonderful lab people, Professor Alessandra, newly minted professor, who's been involved in a lot of this work. Hiro, who's been driving a lot of the work that you've seen here from TY1 all the way down to UX1. Salwa, Josh, Deanna as well, Kara, Jess, my wonderful RAs, and then Anne and Charlotte, who are my interns, my med school bound interns, I hope. Of course, my collaborators, Eugenio and Russell, and all the wonderful resource and support that we get at the Heart Institute that just enable us to do all of this, but also submit grants and publish papers, Gina, Jane, and Cheryl, of course. We thank all the people who fund us, CERM, NHLBI, and begrudgingly named the Department of War. So with that, I'll take any questions, and I thank you for your kind attention. Yes, Lolly.
So call it Department of Defense.
Good. I
would
would.
be too.
Yes.
So I think people like me probably have a lot of basic questions about this biological phenomenon because it's so interesting.
Yeah.
And still new. But one thing just I noticed right off the bat. So one of your slides, I think it was slide 24, you showed that both the IV and the oral delivery showed similar levels of efficacy,
yes.
Right? So, which is amazing. Did you deliver it at similar doses, or did you have to adjust the dose based on the route that?
So that's a wonderful question. So yes and no, and so the typical dose that we give animals is 0.15 mg per kg. And then I read a paper from 1970 something. I don't remember what it was, but it was talking about casein encapsulation, and that 70% of the material in solution was encapsulated. I thought, okay. So I upped the dose to 0.2, so from 0.15 to 0.2, which is ostensibly the same. And yet it's more effective. If anything, in some disease models, it's marginally better. Now, why would this make sense? Here's my rationale, and it may end up being not true, but when you're sending it to macrophages directly in the gut, it's a more directed delivery than it is just injecting it into the blood. A lot of it going to the liver, to the spleen, and elsewhere. That's not to say that there's no macrophages in the liver. I mean, there's Kupffer cells, but I mean PBMCs and monocytes more than anything. So I think that's what the case might be. Yeah.
And so do people in the field think that exosomes from different tissue origins might actually interact with each other? And I'm going somewhere with this because can it act as a if you had tumor exosomes and normal tissue exosomes, would they be in a battle in the body?
Oh, would they?
Could the normal ones overcome the
that's a great question.
ones?
That's a great question. So the concentration of EVs in blood is somewhere along the order of 10 to the 11th particles per cc. A lot of that comes from the platelets that shed EVs on their way from erythrocytes, and then the other ones are from the endothelium. So I don't know if that there's necessarily there's a counter argument against a tumor that's trying to grow. I'm not sure, but in several studies, I mean, I'm not going to lecture it to you, but in several studies, EVs really do prime distant tissue, and if you somehow disrupt that inflammation bridge, then you really do curtail the tumor. It's hard to say. There has been one interesting study about why it is that the heart is so resistant to tumor growth. And it ends up being that some of the microRNAs that are being secreted by healthy cardiac fibroblasts actually inhibit tumor growth. So at least in the context of the heart, there's that happening.
And people think it might have something to do with the contents that's inside of them, or is there anything on the outside?
Yeah, well, so far as they've looked, I mean, remember, this is from the microRNA era where everyone just looks at microRNAs and sees nothing else. They boiled it down to microRNAs. That's not to say that the proteins, and because it takes a whole system to meet another system, the proteins, even what the EVs can trigger on the surface of their target cells. But it is essentially a war of information.
I have a few questions.
Yes.
But I'll try to limit to one or two.
We have time.
How stable is the secretion of these EVs in a long time, over time period?
Yes.
In other words, if I monitor X today,
right,
would that X be there in a week or months or a year from now?
We've never tested this directly, but I'll tell you this. So when we used to isolate CDC EVs, we would wait 15 days. So that means that EVs that were secreted, and we would look at the EV content at 3 days, 5 days, 10 days, 15 days. And the ones at 15 days were much more. So presumably, maybe, there's no real way to definitively say this, but there's an accumulation of these EVs, which means to say that they're probably very, very stable because they're left at 37 degrees in tissue culture. We've tried lyophilizing EVs before, and they're stable there. We've tried feeding EVs to animals using a casein formulation, and that's worked before. EVs are just generally very, very stable, and that's owed to the fact that, as one reviewer says, that EVs are quite crunchy, meaning that the lipid bilayer that they have, it tends to be a lot stiffer than the one of the plasma membrane. Now, as these vesicles get bigger, like tumor oncosome, the stability tends to go by the wayside, but the smaller ones tend to be quite hardy. They're almost the size of viruses, and if you squint, they're kind of protoviruses. They're not, they're replication deficient, but their topology and their structure are similar to viruses, which, they have variable stability, but some of them can be quite stable.
Pointed out that they primarily affect macrophages.
Yes.
Macrophages in different places in our body have totally different functions.
Yes.
They're differently programmed, etc., etc.
Right.
Is there any preference to a specific subset of macrophage somewhere that these guys target?
That's another great question. The theory that we have is that they're being uptaken, especially for IV injection, that they're taken up by monocytes. And when you're giving the drug orally, they are taken by macrophages, intestinal macrophages, but when they're traded to PBMCs in circulating tissue, these are monocytes as well. When we do studies where we deplete monocytes, the effect goes away. So all the cardiac macrophages are still there, but somehow the effect is gone. So we're thinking they're preferentially signaling to circulating PBMCs, and that's not to exclude the T cells or the B cells or the neutrophils or the other PBMCs that are around in circulation, but it just so happens that monocytes tend to be uniquely effective, maybe because we come from a background of acute injury where macrophages are just critical. But there have been papers that I think from Eduardo's lab, showing that EVs can also signal to T cells to skew their polarization more to a Treg leaning phenotype.
If I may, one more.
Please.
For us to be quiet after that. You referred a couple of times to intestinal macrophage.
Yes.
Which brings to me the notion of the microbiome.
Yes.
And whether the abundance of certain bacterial strains within the gut may affect the function. Anybody look into that?
No. Although I will tell you, we have. Yeah, you're raising a really interesting point. So we did it once in one model. So yes, so in a model of heart failure and preserved ejection fraction, these are wild-type mice. We give them a high-fat diet, and then we give them an RNAs inhibitor, and then they develop all these features of heart failure with preserved ejection fraction, which is now the most common form of heart failure. And we fed them TY1, and we want to know, did that shift the population of commensals? And it did. But given the fact that at the time we were searching for a mechanism, we were way in over our heads. So all we did was, and we move on. But invariably, in an orally administered drug, we have to wrangle with this question. In fact, Eduardo emailed me a couple of days asking sort of the same thing. I wonder, how exposure of these, drugs affect the commensal response. It's a funny place. The gut has more immune tissue than anywhere else in the body. And the metabolites that are being secreted by these commensals affect everything from depression to heart failure to cancer to whatever else. It's the Emerald City, but we're yet to get there. We have to get through the poppies first, it seems.
What made you guys think about giving these orally?
I'll tell you because I remember. One afternoon, Eduardo sent an email, and he said, wouldn't it be wonderful if we can give these orally? And I said to myself, challenge accepted. It was during the time of the pandemic, and everyone was feeling a little bit desperate. So I would just read the literature and see what kind of encapsulation methods. Now, Amin, who has done some amazing work with Duchenne, had previously formulated CDC EVs in casein. But casein for RNAs on its own did not work. It worked well for EVs, but it didn't work well for RNAs. So I went to our chemical shelf, and I was looking for anything else, and I saw chitosan, and I said, I
saw that, yeah.
Yeah, and then I went back, and there's that paper from 1970 that said somehow that structurally casein and chitosan are a great match because they form this very strong lattice. So we said, okay. So we mix the RNA with casein in a solution of casein, and then we add chitosan with acetic acid because under acidic conditions they complex. And we were hoping against hope that it would complex and encapsulate the RNA, and it seems like it did. So that's kind of where we're not fancy people. We're very Soviet about our approaches. Simple is better.
I've got a friend and former postdoc who is a chitosan expert. He's made chitosan nanoparticles.
Really?
Interested in continuing that forward.
Please.
Looking for projects.
Yeah, because apparently not every type of chitosan works. It has to be a certain acetylation level. It has to be a certain size, and it's a non-trivial area of that drug development.
Yeah.
Yeah.
So per the question that Zev had posed earlier, the exomers. The exomers were not delivered in exosomes.
Yes.
So what is, when you're delivering those IV, what is the half-life?
Oh, that's a great question.
How long do they hang around? And I guess when you deliver orally, that's kind of a different.
Yes, and these are both questions that we've studied extensively. So, and the two superstars that ran these studies are right next to you. But when they're given, they're formulated in lipid nanoparticles. It's DARM effect, the same sort of transfection reagent that we use for microRNAs. And the half-life for those in most tissue and in circulation is about 24 hours, not much more. And they accumulate quite quickly. Orally, the accumulation is a lot slower, but the bleed is a lot longer. So I think the biggest holdout in tissue was somewhere between 72 and 96 hours. Yeah. So the profile is different, but also the tail is different. The decay is different.
Any other question?
All right, well, thank you guys. These are wonderful questions and wonderful audience. And I hope to keep flirting with cancer for a while.
Good.
Yeah.
We'll keep flirting with you.
Yes. Yes.
All right.
All
Excellent.
right. Thank you guys so much.